Okay, let's talk about the cell cycle. All right, trying to draw a circle there. You can pick any point. I'll start right up there. Um, so I'll label that start. Cells start out small, and then as they grow, they get bigger, and at some point, they're going to need to divide either through mitosis or meiosis. All right, but we're going to take a look in this set of notes what happens as it grows before it divides. All right, so the first phase is called G1 phase. It stands for gap one phase, or you can think of it as growth, because that's what happens. The cell grows and the organelles are copied. All right, and these are like your ER, your Golgi, if it's a eukaryotic cell. There's some checks that goes on in there too. All right, the next one is called S phase, and that's for DNA synthesis. Right, so we're making copies of DNA. So what's important here is that the way DNA synthesizes or copies itself, right? So here's your double strand of DNA, your original strand, and it's going to separate, and then each strand here in blue is going to be used to create another newly made strand, and eventually these two strands will separate in cell division, mitosis or meiosis, but they get copied here in the S phase. Okay, so this has a very important term, and it's known as semi-conservative. Okay, that means each new DNA molecule, like this will become its own and this one will become its own, will keep an original strand, which is in blue, and get a newly made one in red. Okay, so it's known as semi-conservative replication. All right, so let me continue here with the next phase is called G2 or gap 2. And there are some very important checks that happens here. We'll just call them final checks. It's like a checkpoint, final checks. So it checks if the DNA is copied correctly, because if it's not copied correctly, you're going to get all kinds of disorders when the cell divides with improper replication. So G1, S, and G2, they all make up what's known as interphase. I should have picked a different color because I already used blue for DNA. Oops. All right, so this is all known as interphase, and together G1, S, and G2, interphase is when the cell is doing its normal functions, its normal jobs. So like a muscle cell might help in movement, nerve cells might help in communicating, and so on. So that all of that normal functioning happens in interphase. Okay, the final little slice here is called M phase, and that's known as cell division. As the name implies, that's when the cell is going to divide, because at some point it's going to have to divide. And that's either mitosis or meiosis, depending on what kind of cell it's going to make. All right, one last thing I want to add in here is a lot of times you'll see chromosomes. Let me see, where can I add this? Kind of like in that kind of structure. But in the S phase, I don't have room in this chart here, but if you do, you can add it. So this is like G1 phase, chromosome like that, and then during the S phase, when it does this semi-conservative replication, it's going to copy itself, and then you get this sister chromatid looking shape, which I'm sure you're familiar with, and then afterwards, all the way into cell division until anaphase, when these things separate, is going to look like that. So just want to point out that if you see chromosomes in that format, it's before the S phase. If you see it in the sister chromatid format, sorry about that. Our principal interrupted this video. Or if you see the sister chromatid form, that's after the S phase. Okay.